They say an image is worth 1,000 words. So then is a sequence worth 1 billion years? Arriving at Haystack, not knowing much about the lay of the land, a duo had a dream to not just meet the local flora and fauna, but to sequence and uncover the molecular memories these organisms carry. To do this, they helped set up a temporary lab space in a building normally used for ceramics, a minimal viable biolab, or MVB, they called it, a space to get DNA sequences or die trying. They set out to bioprospect. They divided the exploration into two parts, land and sea. They brought tools with them and procured others from the surrounding workstations to collect samples from the local environment. Strolling down to the coast, they encountered a wide variety of species, lichens and moss clinging to trees, seaweed undulating with the waves, kelp that looked like a strange flower, black algae basking on the rocks, pools of red algae teeming with biofilms or sea foams, all containing multitudes. Back on drier terrain, they set out for a mythical meadow, a short walk from Haystack, where they found early signs of summer. With the wind threading through the grass like a slow breath, they discovered flowers blooming, or beginning to bloom, as if rehearsing for the main act. They sampled carefully and respectfully, taking only what they needed. Returning to the lab, they prepared to extract DNA from the samples. Some unwitting critters fell into their sequencing space and became samples themselves. They created a protocol to lyse, centrifuge, and vortex molecules to release their hidden information. But Haystack is often a cold place at night, and many of the reagents and enzymes that were essential for experimentation solidified in these conditions. But fear not, your sequencing team are intrepid folk. They prepared a room where they could better control the temperature, and soon they were able to perform DNA extractions effectively on their samples. But that was only half the story. To read back the sequences of DNA contained by these beings, they brought a portable sequencer with them. It's said that this technology is capable of operating in the field, but was it capable of operating in a haystack? When the room temperature plummeted below the operational minimum, it seemed like the wise man's prophecy would be correct.
I love coming to Haystack because I bring all sorts of things I'm working on, interesting technology, and whatever it is, it breaks. When I come here, the artists find all kinds of ways to use things in ways we never thought of, all kinds of ways to break things, and we learn so much from it.
However, with a little help, they were able to find a solution: the drying cupboard of the ceramic studio, aka the hot box. They turned it into a temporary sequencing closet warmed by space heaters. And hope. Preparing the sequences was long and arduous work, which involved some waiting, and more waiting, and yet more waiting. And once everything was ready, they loaded samples for day-long and overnight sequencing runs, and it failed. And failed again. And then so it seemed like the duo achieved what they set out to do. They peered beneath the bark and petals, rocks and sea. They saw into the spirals of DNA that encode ancient instructions. But this was just the beginning, the first on-site DNA sequencing at Haystack. And now that we know it can be done, the possibilities are endless.